In the previous video, I had shown you the organelles and the structure of a bacteria. All of the bacteria has a layer in between the plasma membrane and the capsule, that is cell wall. Cell wall is one of the most important part of a bacteria, and that's why it is really a good target for antibiotics. In this video, I will discuss the structure, functions, and formation of bacterial cell wall in detail. Bacterial cell wall is made up of sugar backbone and peptide chains. This sugar backbone consists of glycans, these are monosaccharide units, and peptide chain, that is multiple amino acid chains, a pentapeptide. If we magnify, we will see these glycans are attached with each other and form the sugar backbone, and the peptides on these glycans are cross-linked with each other. In this way, the whole molecule of peptidoglycan is cross-linked with each other around the bacterial cell membrane, and this macromolecular unit assembly is called the peptidoglycan layer. There is a space in between the plasma membrane and cell wall that contains a gel-like matrix called the periplasm, and this space is the periplasmic space. This space basically contains various types of proteins that concern with the cell's metabolism. Okay, what are the functions of the cell wall? Cell wall surrounds the cytoplasmic membrane and give the bacteria its real shape. It gives an extra layer of protection for the internal organelles. It helps to anchor the appendages like pili, flagella, and fimbriae, which originates in the plasma membrane and protrude through the cell wall to the outside. It gives mechanical strength to the bacteria, and the strength of the wall is responsible for keeping the cell from bursting out and shrinking, plasmolysis. So when there are large differences in osmotic pressure between the cytoplasm and the environment. So it maintains the osmotic balance. Maintaining osmotic balance is required for bacterial survival. Now let's see how. Osmosis is the movement of water or any other solvent through a semi-permeable membrane from a low solute concentration area to high solute concentration area. This area is highly concentrated with, let's suppose, glucose solution, and this area is less concentrated. These are separated by a semi-permeable membrane. So now to make an equilibrium, the balance of concentration in between both the areas, water from this area will start to move towards here. This phenomena is known as osmosis. And the osmotic pressure is the force required to prevent water movement across the membrane. Bacterial cell wall basically gives the mechanical force to prevent the osmosis. Bacteria, when they're present in human body, they're surrounded by body fluid which is less osmotic than the bacterial cytoplasm. So in this case, bacteria are present in hypoosmotic environment. This is also called hypotonic environment. So due to osmosis, bacteria will absorb water from the extracellular fluid, and as a result, bacteria will start swelling. If no cell wall is present, the uncontrolled swelling of bacteria will eventually cause a burst. Now, in some conditions, bacteria may be present in hyperosmotic environment where the surrounding fluid environment will be more concentrated than the bacterial cytoplasm. So when bacteria are present in hypertonic environment, due to osmosis, bacteria will release water from its cell cytoplasm to the extracellular area, and as a result, bacteria will start shrinking. If there is no cell wall, bacteria will keep on shrinking and lose its shape. Eventually, the cell will collapse and lead to plasmolysis. Okay, now we understand when cell wall is present in bacteria, this prevents the osmotic pressure and protect the bacteria from the osmotic burst or plasmolysis. Now let's see how this cell wall is formed in a bacteria. The peptidoglycans are formed inside the bacterial cell cytoplasm. We have already seen the peptidoglycan is made up of glycans and peptides. Now these glycans are made up of two monosaccharide units, N-acetylmuramic acid and N-acetylglucosamine, in short NAM and NAG. They stay attached together. And the peptides are made up of short amino acid chain. This short amino acid chain stay attached to the NAM of this unit, and this total setup is the peptidoglycan. In bacterial cell wall, the NAM and NAG skeleton is repeated again and again, and the sugar backbone is produced. On every NAM unit, a peptide chain is attached. Now, these peptides are cross-linked with each other. However, a new bacterial cell does not have the complete peptidoglycan layer from its beginning of life. During the growth, the daughter bacteria develops its peptidoglycan layer and make the full cell wall. Synthesis of cell wall. Synthesis of cell wall consists of basically three stages: the cytoplasmic stage, the membrane stage, and the extracellular stage. In stage 1, inside the cytoplasm, the building blocks of peptidoglycans are produced. The NAG skeleton is produced inside the bacteria from glucose molecule by multiple steps. NAG is then converted into UDP-NAG. Here one UTP, that is uridine triphosphate, is utilized and UDP is attached with NAG. Then some of the UDP-NAG will be converted into UDP-NAM, that is the second monosaccharide. This conversion takes place by the enzyme enol pyruvate transferase. This enzyme takes part of molecules from phosphoenol pyruvate and attach them with UDP-NAG and produce UDP-NAM. UDP-NAM is then conjugated to amino acid chains, that is a pentapeptide. Now, in the next step, the UDP portions from both UDP-NAG and UDP-NAM will be released. And this NAG unit will be attached to the NAM unit, which has the peptide chain. Now, this is the peptidoglycan unit that is produced inside the cytoplasm of bacteria. In the second stage, the membrane stage, the formed peptidoglycans will be transferred from cytoplasm to the outside. The peptidoglycan move near to the cell membrane, and there is a protein named bactoprenol protein. This is actually a flip protein. The peptidoglycan binds to the bactoprenol protein. A conformational change occurs in the protein. This flips out the peptidoglycan and releases it to the outside of the cell. The enzyme works here is peptidoglycan synthase. After releasing the peptidoglycan, another conformational change occurs and the bactoprenol proteins come back to its former structure. And the enzyme work here is phosphatase. This is how all the peptidoglycans that are produced are accumulated outside the cell. In stage 3, the extracellular stage, when the formed peptidoglycans are accumulated outside the cell membrane, they start to getting organized around the cell membrane. Two important reactions take place in this stage. Number 1, the monosaccharides, NAM and NAG, of two different peptidoglycans are fused together. Around the whole cell membrane, these glycans units are attached by the reaction glycosylation. And number 2, the peptide chain of one peptidoglycan is cross-linked to the peptide chain of another peptidoglycan. This cross-linking reaction is called the transpeptidation reaction. With these two reactions occurring simultaneously, the real peptidoglycan layer of the bacterial cell is finally synthesized. In glycosylation reaction, the enzyme that catalyzes the process is glycosylase, and in transpeptidation reaction, the enzyme is transpeptidase. This is how the bacteria form its own cell wall. Okay, one important question comes: for how long the bacteria will produce this cell wall? Is it for its whole lifetime? Obviously no, because if the cell wall becomes more folded and larger, the bacteria will become bulky, heavy, and weaker. None of us wants to become bulky, right? Even bacteria do not want to. So when enough cell wall is produced, the bacteria has an auto-mechanism system which blocks the further formation of cell wall. The autolysin enzyme is released from the bacterial cell that block the formation of cell wall and cut the network of peptidoglycan layer to make the bacteria look healthy and smarter. Bacteria is actually a very smart organism. Okay, thank you for watching. I hope you liked the video. Put a comment if you have any question, and for getting more videos, click the subscribe button of this channel.